Thank you, Praveen. Welcome. Hello, everyone. I'm going to talk to you a little bit about some work I have been doing for the last 2 years. And essentially, it's looking at estimating plant pasture biomass and quality from UAV-borne, or drones, as we commonly call them, imaging across Queensland's rangelands. These are areas of natural pasture lands across Queensland, which is a very large state in Australia. I'll talk a little bit firstly about some existing and potential methods. Many existing methods are used across the rangelands, from exact precise measurements to inferred estimates, including manual harvesting, rising plate estimates of pasture height, proximal sensing, such as sonar, terrestrial laser scanning, phenological camera modelling, the UAV photogrammetry and LiDAR, hyperspectral time series estimates of plant fluorescence, fecal NIR sampling, and both field and UAV-based hyperspectral imaging, each having unique benefits and challenges. The purpose of this piece of research was to develop a method to estimate plant pasture quantity and quality that is comparable to traditional manual sampling techniques as a more efficient, accurate, and scalable means of mapping the pasture resource, as well as providing new opportunities to build or fuse into satellite-derived imagery, and importantly, as a standalone measure of the current pasture state for more sustainable stocking rate determination. We start with the study area. This is a broad stratification of both wooded and non-wooded landscapes that are further classified into arid and tussock or Mitchell grasslands and savanna and subtropical woodlands. A total of 19 sites across each land type and vegetation type were sampled. This is an example of the four broad vegetation categories that we sampled. The top left is the arid grasslands. Top right, Mitchell or tussock grassland. Bottom left, a subtropical woodland. And bottom right, a typical savanna woodland. At each of these field sites, a range of measurements and samples were collected using a star transect method. Along each of these sample transects, 18 biomass and rising plate height quadrats were established. Samples of biomass were weighed to determine the total standing dry matter, as well as chemical analysis to determine both crude protein and acid detergent fibre as measures of both yield and digestibility. Alongside each field measurement was a UAV campaign, starting with photogrammetry and hyperspectral sensing. To begin with, the photogrammetry involved ground control, which was carried out with differential GPS that was coincidentally processed through a base station when in remote locations. A benchmark terrestrial survey was carried out to determine the sub-metre accuracy of this system. A high-grade digital SLR camera was used to capture a dense coverage of red, green, blue imagery, used to produce both an orthomosaic of the canopy and a height model of the pasture. In addition to the photogrammetry, a scanning hyperspectral sensor was used to capture visible near-infrared imagery using a Corning Shark push-broom sensor that collects 150 discrete bands from 400 nanometers to 1,000 nanometers. Three main processing streams were developed. First, the hyperspectral data involving the orthorectification, radiometric calibration, and regression of spectra with field sample pasture chemistry. Photogrammetry involves structure from motion techniques to derive a pasture height model and robust regression with field data to develop a model of total standing dry matter predictions. The results of the pasture height field measurements and the derived canopy models are presented here for each land type. The developed pasture biomass model from the relationship of height to mass for the broad categories of grassland and woodland were developed through a robust regression modelling approach that was used to develop predictions of total standing dry matter and compared at a field test site. Next, the acid detergent fibre chemistry was modelled using all spectral bands to determine the best indicator. Acid detergent fibre is a measure of the least digestible part of a plant, in particular the lignin and cellulose layers of a plant cell. 651 nanometer band was then used to make model predictions across two sites of what appeared to be different historical grazing intensities. A lighter graze site in a reserve and a heavier graze site closer to stock water. Acid detergent fibre was higher at the heavier site, potentially indicative of more unpalatable pasture species. Next, crude protein as an indicator of pasture quality was mapped in the same manner. The red edge to near infrared band 759 nanometers showed the strongest relationship with the field chemistry. Differences between management didn't appear as distinctive as the acid detergent fibre, as potentially a function of similar low protein native pastures and or limited by the constraints of the limited number of samples in the model development. This is a proof of concept and shows great potential. Further research and potential use of the shortwave infrared portion of the spectrum is recommended, but still a costly approach. Both field and UAV measurements are noisy and difficult to quantify precise error. More calibration and validation of both biomass and digestibility mapping is needed. However, management effects can potentially be seen at the pasture sort or patch scale, showing great potential for data fusion and as a standalone paddock or property scale mapping tool. This work has recently been published in the NDPI Agri-Engineering. Provide some acknowledgements of the funding body and of the various properties that provided access and support for our field sampling. And the people that I worked with at the Queensland Department of Environment and Science, University of Southern Queensland, University of Queensland, and Department of Agriculture. Further research is ongoing and currently underway. Terrestrial laser scanning and field spectroscopy along with a fixed-wing UAV, is allowing us to broaden our research, as well as testing the sensitivities of the system at known locations for the purposes of modelling changes through time. Thank you, everyone, and I invite some questions.
Thank you, Professor Jason, for your presentation. Yeah, it's very informative, and I will see if anyone have any questions or comments, you can please go ahead and ask Professor Jason.
Hello, yes.
Yeah, Professor.
Can you hear me? This is Yayun Wang.
Yes, go ahead.
Can you hear me?
Yes, go ahead.
Yeah. So in your title, you said this imaging method also can visualize the quality of the pasture. And how I'm not quite sure, or I didn't get it. How does the imaging can monitor the quality of the crops?
Okay, sure. So quality is inferred from two indicators, crude protein and of digestibility.
Okay.
So we infer quality from those two measures of both pasture nutrition and pasture digestibility.
Okay, thank you.
No problem.
Thank you, Professor.